There it goes. Oh, oh, whoa, that was wrong. There we go. I did the wrong thing there. So this will be posted, this video should be posted up online sometime this weekend or something like that. But we have just finished a section. We have had a test. It is still way too hot in this room. What's that? Yeah, let's go lecture in the rain. I'll try to take the projector with us. It's brand new. Hopefully the rain doesn't. They're all waterproof now, right? That's what they do now, right? How long do you think? Yeah. No.
I don't think the heat's on because we have air conditioning back here. So we just switch the password. Seriously, you can just stand back here.
I'll lecture from back there. I like that idea. Anyhow, how long do you think it is before like your phone has a projector in it and you can just like take it out, set it up like this, and be like, there we go. That'd be awesome. Can you imagine movies done like that? Like, hey, I want to watch a movie. I'm out in the middle of nowhere. Let's just project on the side of this building here. Okay, never mind. Okay, so let's talk about the next section that we're moving into. We just talked about cell membranes. Now we're going to talk to essentially a subset of that topic and talking about specific types of proteins and specific kinds of functions of receptors, especially getting signals across a membrane. So I'm going to walk back up here towards this putative air conditioner. We're going to talk. But as I do that, let's talk. Let me just back up a little bit and talk kind of generally about. Oh, that's. Let's just talk generally about. Reflect for a moment on most of the life that you know. Most of the life that you know is multicellular. Now, that being said, there are a lot of bacteria all over and in you. You need them to live. They could make you sick if they get out of whack and all those other things. And so you are, even if you don't know it, intimately acquainted with unicellular life. But most of the life that you know and you interact with, that you put in your mouth and chew up and eat, plants or animals or fungi or whatever else it is, other people that you interact with, your pets, all of that's multicellular. I guess that's probably not coming as a surprise to you. But in order for this to happen, in order for multicellular life to exist, these cells need to learn how to work together. You can't just have some cells going off and doing their own thing. That causes things like cancer and whatnot, and that's bad. So cells need to somehow learn to cooperate. And in order to do that, for cells to work together, they have to be able to communicate. You can't cooperate with something that you cannot exchange information with in one way or another. Either information that has been exchanged before you cooperate, some sort of predetermined signals, or as you are cooperating, but you need to have information passed between two things that are going to cooperate. So what we are going to talk about now is how that happens. How do cells communicate with each other? And particularly, we're going to be focused on, just for a moment, how the receiving cell receives that signal and how it gets turned into some sort of response. And this is a process known as signal transduction. Okay. So let's just begin and start talking about the nuts and bolts of the nuts and bolts of this, the components that go into it. First off, one thing that you need is some sort of protein receptor. This is going to be a protein on the cell surface that will bind to a ligand, a signal molecule of some sort, and actually do something with that. This is usually a glycoprotein. Not always, but usually a glycoprotein. If you have a receptor, it kind of follows that you would have something to be received. And that, in this case, is a thing known as a ligand. A ligand is pretty simple. It's kind of a fancy word for something simple. It's just a molecule that's going to bind to a receptor. And then has some sort of effect. Many of these are things known as hormones. Hormones particularly are signals that are going to act in you at a distance, usually not at a cell right next to them, but a cell somewhere distant in the body. So together then, we have this ligand that's going to bind to a receptor and becomes this thing known as the ligand-receptor complex. And this ligand-receptor complex is going to in some way alter cellular function. So because of the existence of this, the cell will not be the same as it once was. This can take the form of a lot of different things. It's not just necessarily a narrow scope of things. It can really be a lot of things. So for instance, some things that this could be is something like gene regulation. We could be turning on, up, off, down certain genes, the expression of certain genes. We could be altering enzyme activity. We could be increasing transport of something across the cell membrane. So there's a lot of different examples of things that could change in the cell. These are three of the most common things that happen, but this is by no means an exhaustive list. So let's go back to hormones just a second. I want to give you a more precise definition for what a hormone is. And really, hormones are a blood-borne molecular signal, or ligand. And they are produced in one particular, some sort of cell, but then they bind to another cell and end up affecting something in that other cell. Some of the kinds of things that we just talked about, like gene regulation, enzyme activity, transfer across its membrane, something like that. So really, these hormones can fall into several classes, or at least two classes. One major one is the lipid-soluble hormones. Primarily, these are steroids, but not exclusively steroids. There's a few others as well that we'll talk about. Now, the interesting thing about this is, so if you're a steroid and you're lipid soluble, how would this be different than something, say, a hydrophilic ligand? Yeah, Christian.
It can go through the cell membrane?
Yeah, this can pass right through the cell membrane. And they do. So these steroids generally pass straight through cell membranes. Aren't really interrupted by them at all. And then they bind to a receptor that's inside the cell. So most lipid-soluble hormones have intracellular receptors. They're not sitting on the cell surface. They're actually sitting on the inside of the cell. And the cell membrane actually provides kind of a little bit of a selectivity filter that only allows certain kinds of hormones to get through, particularly the lipid-soluble ones. So let's talk about a few characteristics of this, a few characteristics of lipid-soluble hormones. Number one, so the lipid-soluble, this hormone receptor complex that's going to be made once it binds to the receptor is going to enter the nucleus and then alter gene expression. This is the primary way in which they act. What this means is because you're changing gene expression, lipid-soluble hormones tend to have very slow responses. These do not happen quickly. This is not a kind of hormone like some others, like beta-adrenergic hormones or something like that, that's going to very quickly change how a cell functions. Because you're changing gene expression, there's a lot more steps till you actually see the end result. And so it's very slow to turn on. However, that being said, it also has a very long-lasting effect. Because you're actually changing gene regulation, the proteins that you create from that will could be around for a significant amount of time, and therefore the effect of this is very long. But this is kind of efficient because if you need a long-lasting effect, you can just give a few signal molecules and you'll have a long-lasting effect. Whereas some others, you give it to a cell and the signal turns out very quickly, and so you have to keep applying signal to get change to happen. Kind of to illustrate how this works, so here we go. We have this lipid-soluble hormone. It is in the bloodstream. It very easily diffuses out of that. In fact, it kind of wants to, because being lipid-soluble, your blood is aqueous, in case you haven't noticed. Well, at least it's aqueous until you go down to iceberg and have 5 hamburgers, and then it becomes more of an oil consistency. But other than those instances, it's an aqueous solution, which means that these lipid-soluble hormones are not very easily dissolved in there. You generally have proteins that are binding to those lipid-soluble hormones and taking them out, but it also means that they're very readily going to diffuse, get out of there. They do not really want to be in this aqueous solution, and so they'll very readily move out of the bloodstream into the surrounding tissue, straight through the membrane of surrounding cells, and come and make this receptor hormone complex on an intracellular receptor, which moves into the nucleus, does something with, which can be quite a few different things. Can do something interacting with the genome, change the gene expression, and then you get a change in the amount of protein down here somewhere that's being made, changing the effect of the cell. So a couple examples of this. We have talked already about hormone structures, and this should be somewhat familiar to you. This is testosterone. It looks very, very similar to estradiol, which is the major estrogen found in humans. Another one is cortisol. It has a little bit extra doohickey up here. That's not an actual functional group. No good, Dr. Brannigan's. What is this functional group? It's a doohickey. You probably won't get points for that. I don't know if he gives out creativity points. But if you do, you have to give those to me because you copied me. There are some other lipid-soluble hormones that are not steroids. One example of that is something called Thyroxine. This is also known as T4 because you have 4 iodines on it. This is like the major place. Like, you need to get iodine in your diet. Like, the only place that your body uses iodine is right here to make thyroxine. It's not exactly, but it's really, really close. Like, 99.9% of all the iodine that your body uses gets used in this one molecule. It actually normally only has, in its inactive form, it has 3 iodines, and then it gets an iodine added onto it, and then becomes active, going from T3 to T4, and becomes more active. Anyways, long story there. But there's also another lipid-soluble hormone, and so they're not all steroids, but majority steroids. Another major class, yes?
Do you want us to know those, or just be familiar with those?
You should be familiar. Like, for instance, if I gave you this and asked you, is this a lipid-soluble hormone or a peptide hormone?
Yes.
You should be able to let me know, yeah, which one of those it is. But that being said, I mean, I won't say, give me the structure, draw me cortisol. And if you miss one of those OHs, it is all off. You might as well just drop the class. No, like, I want you to be able to say, if I say, hey, here's a hormone, is this going to, or let me give you another one. I give you a hormone and say, this. And I say, where is the receptor found for this? In the cell membrane or intracellularly? You should be able to tell me it's an intracellular receptor. You should be able to recognize, hey, we have this ring structure that's characteristic of steroids. Steroids are lipid-soluble hormones. It's probably going to have an intracellular receptor. I can also ask you, is this more likely, give you this hormone and say, is this more likely to have a short-term response or a long-term response? You'll be able to tell me it has a long-term response. So I'm not asking you to like reproduce these, but you should be able to look at something and say, is this probably what class does it fall into? Does that make sense?
Yeah.
Okay. Okay, good question though. Peptide hormones. This is another major class of hormones. As you might guess, they're made up of amino acids. These can be proteins, usually very small proteins. they can also be amino acids or amino acid derivatives. So some modification on an amino acid. So let's talk for a few minutes about how these act. So, do you think these can go right through a membrane? No. So amino acids, they're hydrophilic, they're polar. They don't enter the cell, but they're going to bind to cell surface receptors. So they're going to bind to some receptor on the cell surface and have some effect. And each is going to have some specific receptor that determines how the cells are affected and how. In fact, most of these not just have a specific receptor, they generally have lots of different specific receptors that are kind of cross-reactive to similar things. That's a little bit more than you need to know at this point, but it's kind of interesting how that works. So something like epinephrine, major component in adrenaline. Actually, if you go to the UK, like epinephrine is just called adrenaline. And anyways, it has like 5 or 6 different kinds, major classes of receptors that it has throughout the body, and each of them have slightly different effects depending on what cells have which kind of receptor. Now, once you have the peptide bound to a receptor, there's a couple ways that it can then affect the remainder of the cell and actually carry out the work that it needs to do. And one of these is that you can have a G protein and second messenger system. So the hormone itself is the first messenger. It can't enter the cell. It binds to the receptor. So right now at this moment, I'm just going to give you kind of an overview of how this works, and we're going to get more into detail on this in just a few minutes. So this is just kind of like the broad brush strokes of how this works. So you have a hormone. That first messenger binds to a cell surface receptor. It's not going to be able to go into the cell itself. However, that receptor is going to activate another membrane protein called a G protein. Talk about why it's called a G protein for a minute. Then the G protein transmits that signal to another membrane protein, which is known as the effector. This is going to be what's actually carrying out the end result some way. And that protein produces the second messenger. And that second messenger then is the message inside the cell. So this way you have a signal molecule now that is inside the cell, but nothing actually physically entered the cell. It is now this second messenger that goes on to actually have the effect inside the cell. Okay, so G proteins, this is actually the most common messenger system that is in animals. It's a little bit different when you get into plants, but in animals, a G protein second messenger is the most common message system that our cells have. And actually, there's tons of diseases that are associated with this system. Dysregulation in somewhere involving a G protein second messenger include things like seasonal allergies, hypertension. Actually, the list goes on and on and on. And actually, 40% of all superscription drugs, that should be prescription drugs, but superscription kind of sounds cool. I don't write prescriptions, I write superscriptions. So stupid. Anyways, 40% of all prescription drug effects actually target G protein systems. You are going to modify a G protein system somehow. So these are really common, really important. Just to also kind of give you just how big a part of biology, especially animal biology, that G protein systems are. Actually, so there's like 3 Nobel Prizes that are going to cover what we're going to talk about today and tomorrow. So the first one, Nobel Prize in Physiology, 1971. This guy, this is Earl Wilbur Sutherland Jr. And he actually discovered a beginning of this. He actually looked at epinephrine and found that it would stimulate the breakdown of glycogen in liver. And then, but did this through this cyclic AMP cascade. So he actually elucidated one of the first second messenger systems, one of the first G protein second messenger systems. And he got a Nobel Prize for that. Then you had these guys, which, by the way, I really like this like 90s background. It's all about. So the guy on the left with the cool 90s background going on, looking like he's in space or whatever that's supposed to look like. This is Martin Robel, and he's actually the one. So Sutherland showed this system, but he actually showed that this was a G protein involved system. The guy on the right, this Alfred Gelman, discovered what proteins were interacting with that. And then just a more recent one, Nobel Prize in Chemistry in 2012. The dude on the right is Robert Lefkowitz, which maybe I should have him on the left. And then we have Brian Kobilka. And they actually, quite a while before the Nobel Prize was given, looked at the second messenger system for beta-adrenergic receptors, which is one of that receptor system is so crazy common and used in so many things, especially things that go wrong in you in human systems. So, like, just this general line of knowledge has produced a lot of Nobel Prizes, and the reason isn't just because they discovered things. They discovered things that ended up being kind of like central to a lot of stuff in biology. So, why do we call them G proteins? And the G protein part of this comes down to you actually have a guanidine nucleotide, so either a GTP, a GDP, or guanosine, binding to the protein at some point, and that forms a heterotrimer. Well, the whole protein forms a heterotrimer. They all tend to be heterotrimers, meaning that you have 3 subunits, and each of those subunits is different. Now, those subunits in humans are the alpha, the beta, and the gamma subunits. So, in humans, you have 20 or so different alpha subunits that you have genes for. There are 5 different beta subunits that you have genes for, and 12 different gamma subunits. And these get mixed and matched in different ways. So you can imagine that 5 times running, there's 100 different combinations that you can make there, and then 100 times 12, that becomes 1,200 different possible combinations that you can make from those 3 different genes. So there's a lot of potential for a lot of different kinds of G proteins. So let's talk a little bit more about the construction or how these G proteins are put together. So first off, as I've talked before, they get their name G because they tend to have a GDP attached to it. And a ligand, yes, Dagan.
So if it's a heterotrimer, does that mean it has to have an alpha, a beta, and a gamma?
Yes. Each protein has an alpha, a beta, a gamma subunit.
Okay.
Yes. So the ligand is then going to bind to the G protein coupled receptor, which is always a 7-pass membrane protein. And then it has on it, generally on the intracellular side, this high affinity site for the G protein. So as soon as you get the ligand attaching, I'll pause it just for a second. So as you get the ligand attaching to the G protein coupled receptor, it then binds to the G protein, and the G protein undergoes a conformational change when the G protein coupled receptor is bound to it. The GDP unbinds and a GTP binds. And then the alpha subunit is hard to live with. It really doesn't like GTP so much. So as soon as GTP binds, the alpha unit says, I'm getting out of here, I just can't stand GTP. And so it unbinds from the beta and the gamma section of the protein, and from the receptor, it kind of goes off and does its own thing. Once the alpha subunit goes rogue, unshackled by the fetters of those that would keep it down, it goes on to modulate other things in the protein, these other effectors. Turns them on or can turn them off. We'll come back and talk about what those do in just a moment. But once alpha does that, it realizes it didn't like them so much anyways, it's kind of lonely for the rest of the G protein, and then it rebinds with the beta gamma. So the alpha, while it's out there doing its own thing, it can have a couple effects. First off, it can either activate or inhibit the effector. It can turn it on or it can turn it off. And what this means is that one hormone, it may activate one particular effector and it may inhibit another effector. It all depends upon which receptor the G protein is bound to. So, just as kind of an aside right here before we keep going, this is something, this is kind of hinting at a principle that happens a lot in physiology. The same molecule does not have the same effect everywhere, especially when we're talking about cell signaling. Particularly, we talked earlier about beta adrenergic receptors. There's beta adrenergic receptors, there's alpha adrenergic receptors, and what can happen is, in some places, let's imagine here smooth muscle. How does smooth muscle respond to epinephrine? Well, in some places, like smooth muscle around your blood vessels, it causes increased contraction of those muscles. This increases your blood pressure. So in places around your blood pressure, epinephrine will cause constriction of those muscles. In your gut, however, it has alpha adrenergic receptors. That same molecule, epinephrine, will bind to those and cause relaxation of those muscles, a completely opposite effect. Even though it's still smooth muscle, and we're still giving it the exact same hormone, it can have completely opposite effects. All of that has to do with just a different receptor and then a different G protein messenger system. But in the end has completely opposite effects. And this happens in biology. One of the cool things about this is that this can coordinate very complex responses in your body by just having different receptors in different places, different messenger systems. This really works actually nicely because if you think about it, like epinephrine is kind of a fight or flight messenger. You need to coordinate a very nuanced response if you're getting your body ready to like run away from bears or something. You do want your blood pressure raising so you can efficiently deliver blood to places. You are not so concerned about digesting lunch at that point. It's not really high on the priority list. So we're going to turn off your, like the peristalsis going through your intestines, because that's not so important. And you can do both of those functions with the exact same molecule. We only need to give one signal, and we can have these coordinated responses that can be completely opposite, but really appropriate in their different contexts. And this is just something that comes up a lot in biology, that you have a very nice balance and nuance to different sections of biology simply by switching out a few different proteins here or there, a few different receptors, and completely changing the effect. And I'll come back to that again because there's things about this. It might look weird and complicated, but there's aspects of this receptor system that make that possible that you couldn't if it was a simpler system. So let's talk a little bit about how this is going to happen just for a moment. We have our ligand up here. This is our G protein coupled receptor. Remember that our G protein coupled receptor is this 7-pass protein. So we can 1, 2, 3, 4, 5, 6, 7. We have seven passes there. And this binds here, and we're going to get a change out of GTP for GDP, and that causes this alpha subunit plus the GTP to unbind and go off and either activate or deactivate some effector. And that effector then goes off, like I was talking about earlier, and actually produces or releases the second messenger that gets the signal going on the inside of the cell. So let's talk about one of these in particular. Okay, yes?
So if the alpha subunit is the one doing all the affecting, what are the beta and gamma for?
They also go off and do affecting as well. So that's kind of, they also do other affecting. We tend to talk about the alpha more because the effects of the alpha were discovered first. And so it's kind of the canonical flow of information is from the G protein coupled receptor to the G protein, then the alpha subunit to the effector. But actually they both have effects. And they have different effects. And actually they can each affect different. So alpha does not only interact with one protein, it will interact with multiple effectors, so you can actually get this kind of spreading effect going from this to many different things. Does that make sense?
Yeah, it's not complicated.
It is. But we're going to distill it down and only look at certain select pathways. And one of them that I do want to talk about is, I'm going to go through 4 major G protein second messenger systems, just to really quickly list them out to you. The adenylyl, cyclase, cyclic AMP. I'm going to talk more about these in depth here in just a moment. We also have the calcium channels and calcium. Calmodulin is also in there. Phospholipase C and IP3. And then also the 1,2-diacylglycerol, second messenger system. Okay, so let's briefly run through these. Well, not so briefly. We'll take them in stride. So let's start with the adenylyl cyclic AMP. So some abbreviations. AC. I'm just going to use to talk about adenylyl cyclase. CAMP or CAMP for this is cyclic adenosine monophosphate, or cyclic AMP, or just CAMP. And then PKA, which is protein kinase A. Can anyone guess from the name what it does? What's that?
Phosphorylation.
Yeah, phosphorylation. What do you think it phosphorylates?
Alpha.
Protein, yeah, close. If you call the protein alpha, it would be alpha. It's alpha because we actually have lots of protein kinases. There's the alpha, and then there's the beta, and the gamma, and the anyways. But yeah, it phosphorylates a protein. That's not very specific. We'll talk more specific in a minute. And this is cyclic AMP, so it looks like ATP, except for, so you have your adenine right here, plus the ribose makes that adenosine, and then we have one phosphate attached onto there, but unlike normally how we have just attached up here through the oxygen to the number 5 carbon. It's also bound to this oxygen down here on the 4 carbon, or 3 carbon, excuse me. So that makes it cyclic. So cyclic AMP. And again, I won't ask you to recognize, or I won't ask you to reproduce this, but you should be able to recognize it. Okay, so let's talk specifically about a adenylyl kinase or adenylyl cyclase cyclic AMP second messenger system. So again, just like before, this is the beginning of this is just a standard G protein second messenger system. We have the ligand that binds to a G protein coupled receptor, which then interacts with a G protein. GDP is replaced with GTP, and the alpha subunit goes off and does its own thing. It elopes with the GTP, which then binds to the effector in this case, which in this case, which is adenylyl cyclase. Adenylyl cyclase takes ATP and makes it into cyclic AMP. So you have an ATP gets turned to cyclic AMP. In this case, cyclic AMP is the second messenger. So this is the message on the interior of the cell. The first messenger is this. Whatever was on the outside that bound to the G protein coupled receptor. The second messenger is the AT or the cyclic AMP that you have right there. Once you have cyclic AMP created in the cell, it's free to go off and do its work. In this case, it goes on and it interacts with protein kinase A. Protein kinase A then does what it's supposed to do, phosphorylates a protein, in this case particularly an enzyme. So we're going to phosphorylate an enzyme, and then that enzyme does what enzymes do. It converts some substrate into some product, and this product then is the final effect of that messenger system. It's kind of complicated, and it has a lot of steps. Are there any questions on that? Yeah, Dave.
Can we find this diagram in our book?
No, you cannot find this diagram. I will upload this diagram plus all the ones I'm doing in this up to D12. This is a diagram of my own sit down and creation. Well, partially, I modified it for a few things. Modified it from some Wikimedia Commons diagrams that were there already. Really good. Okay, so here's just an aside. Wikipedia is a great resource for this, including SVG diagrammatic images where they're vector files, so I can rearrange things and make new things from them. It's great for that. So I think what I did is I got all the G protein this, and then I had to make all of that. But anyways, and I think I had to make the, I don't remember what else I made. I made part of it. Part of it was typed from Wikipedia. Anyways, yeah. I'll upload that. Now, let's talk about a couple things that make this really useful. So we have all these steps back here. So let's count them. We go from one messenger here to at least one G protein, which then that alpha can do multiple adenylyl cyclases. Adenylyl cyclase converts multiple cyclic AMPs. Each of those cyclic AMPs does 1 pK protein kinase A. But then protein kinase A can phosphorylate a lot of enzymes, and then that enzyme can convert a lot of product. So at each step along the way, each thing is reproducing more than itself. It's converting more of the next step than it is. So we get amplification all along this. And this is one of the nice things about having a signal cascade. There are several different advantages to this, but one of the major advantages is signal amplification. So let's just take a look at this adenylyl cyclase cyclic AMP signal cascade. What are the amplification that goes into it? So first off, for every first messenger that binds to a G protein coupled receptor, oftentimes we'll get somewhere in the neighborhood of about 1,000 cyclic AMP made and binding to pKa. So we get about 1,000 of that cyclic AMP pKa complex created. That's a lot. We've increased 1,000-fold. However, that's not all. For each one of those, we get about 1,000 enzymes phosphorylated. Because again, pKa itself is an enzyme, and it's not consumed over the course of its reaction. It can continue to just keep doing what it's doing and make more of it. So it will convert about 100 enzymes before the cyclic AMP that associates from it, and that signal gets turned off again. So for every enzyme, then, about 10,000 product gets made. Now that being said, this is really general. This is kind of a roundabout estimate somewhere in the middle. It's an order of this could be an order of magnitude or two in either direction, depending on what specific enzyme, what specific products we're doing. This is kind of general. But all that being said, you multiply all of these together, and that means that you're getting somewhere in the neighborhood of a trillion-fold amplification. From one single, so for every given one protein that ends up binding to a cell. Yes?
Billion or trillion?
Oh, billion, yeah, my bad. I went from million. That's one trillion. Ah, yeah, no billion. Thank you. Me and numbers. Let me give you an example of this. In animal physiology, I let students do projects there. They get to design their own projects and go and actually carry it out. And one group of students wanted to look at the effect of estrogen, environmental estrogen on goldfish and how that affects their metabolism, right? So that sounds cool. I let them do that, and I order in a gram of estrogen, estradiol specifically, and they proceeded. I told them, okay, so go ahead and figure out what would be like a normal amount, like a normal concentration that you might find, like maybe in the environment if you had like some contamination, or might be normal like in the blood of a human or something else like this. And they kind of skipped over that step. And they went ahead and they put in, I think, 0.1 milligrams. Something like that. Which is actually about as low as they could measure, and then they put it into like 10 mils, and then put that 10 mils into the thing, but that was like the least amount they could measure out, but they didn't want to do, you could do serial dilutions to get it down for that, but anyways. And they come in the next day and all the fish are dead. Are just gone. Okay, so I'm like, okay, this is what I want you to do. I want you to go back and measure, like, figure out how, and this I don't ever know how to word this right, where it just doesn't sound weird and horrible, but it's like a good like measure. Like, okay, if you were to like extract all the estradiol from, how many women would that like have the equivalent of that much estradiol, right? If you said men, it'd be a lot more. How many women would that be? Just to give them a concept, and they did that. So it's pretty easy to do. You find out what is normal concentrations in a human and a human female. And I asked them to go, hey, let's go conservative. So go like the most. Like this is like the one with the most estradiol on the normal end before it gets like crazy high. And then and then you can calculate. So that's blood concentration. Okay, let's calculate blood volume for a human. So you can calculate out the blood volume for a human. And then you calculate out. Okay, so then that's how much you have per human female, and then how many would you need? Actually, turns out that how much they put in there is somewhere in the neighborhood as would be contained in something like 3,600 women. And so if you do that, you'll kill fish. And actually, the whole gram is like 400,000. Like that's like the equivalent of estrogen is like 400,000 women. It's just crazy. Anyways. Long story short, hormones act in really, really small quantities. They have really large effects in really tiny quantities. The reason that they can do that, the reason that you can have these insanely, like, and actually, like, sex steroids are, like, really pretty powerful and potent. But you get that because you get this incredible amplification. Even when one single molecule binds to a cell, it can have this incredibly high amount of signal output. And that's one of the advantages of using this. What that means then is like you don't actually have to use a lot of resources making hormones. You don't have to spend a lot of energy on that. You can just make tiny amounts and send them out into the world being your body, that sounds weird. Send them out into the world and they have their effects. And it's really, really energy efficient. You don't have to make much, and it works really well. Anyways, with that, enjoy your weekend, and I will see you on Monday.